So steroid hormones, they are all derivatives of cholesterol, and they're lipophilic or hydrophobic. They're lipids and they don't like water. So here are some examples. This is actually a pathway of a metabolic pathway that you don't need to know, but I think it's helpful to see these examples. So cholesterol is the derivative of all of these. You get cholesterol from your diet. It's actually important. And there's this pathway, and these actually down here, these 4 final products, there's actually way more than this. And it's this, you can Google metabolic pathways of steroid hormones and you'll see a lot. But these are 4 that we will see. So when you think of steroids, you might think of testosterone. This is a steroid produced primarily in the testes of males. That's who has testes, but also some in the adrenal gland. And estradiol is kind of the counterpart that we think of as being important in females. It's an estrogen, but it's actually also present in males. You can actually see testosterone is metabolized into estradiol. They're very similar in structure. So these are sex steroids that we'll see when we get to the reproductive system. Cortisol, you may have heard of. It is a stress hormone, response to stress. So it's produced by your adrenal gland as well, and we'll talk about it next week. Aldosterone is involved in water retention and kidney function, so we'll see that there. It's also involved with the stress response as well. These are 4 examples, and actually the 4 that we'll see the most in this class, I believe. So they're hydrophobic. They can cross the cell membrane. This also means, I want to show what that looks like going into the cell in a moment, because that's the most important part to know about them. The other thing just to consider is that when these, let's say we've got cortisol circulating in the bloodstream, because that's what hormones do, right? It's not very happy in the blood, just like hanging out. Why not? The blood is mostly water, the plasma portion of blood. So it needs a carrier protein. All of these do, to be happy traveling in the blood. So you'll see sometimes with blood values of like clinical values of steroid hormones, as well as thyroid, there's bound and unbound values, whether it's bound to the carrier protein or not. So let's look at what these hormones do when they bind to a receptor, and they're going to bind to a receptor inside the cell. So I'm going to draw you a lovely cell that has a lovely nucleus. And of course, there's DNA in our nucleus. And let's do a few different colors here. Colors, here we go. Let's do a nice green for our receptor, because it's going to mean go. Not always true, but so here's our receptor. When the steroid signals the cell, it's going to bind, actually enter through the plasma membrane. It can do that, right? Because it's hydrophobic. Let's say this is estradiol going in here. This receptor is going to translocate or move to the nucleus. So let's just choose another color here, nice pink. This receptor is now a ligand bound receptor, a hormone bound receptor. So I'm actually going to label this thing. I'm going to call this, the same color here, a steroid receptor complex. It's going to act as a transcription factor. It is a transcription factor. What that means is going to bind to the DNA and is going to help initiate transcription. DNA being copied into RNA. So we're going to have RNA. What's going to happen to that RNA? It's going to be made into protein. The steroid receptor complex isn't involved in that, but it initiated this pathway. And ultimately, we are going to make a protein. What protein this is depends on the steroid receptor. The steroid depends on different receptors, depends on all kinds of things. There's each steroid receptor complex can act as a transcription factor on multiple genes. So it gets really complex. So we've got a new protein. That's the point here. This is a mechanism of action you might make for cortisol, proteins involved in immune, actually in this case, suppression, metabolic changes. For testosterone, you might make skeletal muscle proteins, skeleton growth factors, kind of stuff like that. While steroid hormones are derived from cholesterol and then metabolized, broken down by enzymes in our body, peptide hormones are actually synthesized from DNA in our body. So it's a very different mechanism of production, in addition to the different mechanism of action. So here's 3 examples of peptide hormones, again, of different sizes. They're all made up of more than one peptide. So they're called peptide hormones, polypeptides, and can be proteins. That's if they fold into a tertiary structure. We're not going to worry about those details. Basically, they can be different sizes. They're all polypeptides, amino acids linked together. So it should make sense that they are synthesized from your DNA. So this is the synthesis process. Your DNA is inside your nucleus, of course. I don't have that drawn right here. This is the process. So DNA is transcribed into RNA. This might be initiated by a steroid hormone. It's a process that happens in your body all the time. So this RNA is then going to be made into a protein. So we've got DNA, RNA, protein, just like usual. Now, this protein is going to be, and that happens in the rough ER with ribosomes, that translation into a protein. This protein is going to go into a transport vessel. It's going to be exocytosis. So this transport vesicle is going to contain, what is this little thing right here? I'm actually going to make it a different color in here so we can track it. Once it's a protein, I'm going to make it green. This is our little protein inside our secretory or transport vesicle. The Golgi apparatus is going to prepare it for secretion. So it becomes a secretory vesicle there. Don't need to know that. Besides, the point is you should remember the Golgi apparatus is involved in vesicle secretion. So that makes sense. And that protein, polypeptide, can then be exocytosed, secreted outside of the cell through the cell membrane to go into the bloodstream and have its effects. So what's really cool is these peptide hormones are encoded by our genes. So you have a gene for the vasopressin hormone, for CRH, for ACTH. These are all ones you'll see. And so they can be regulated whether they're produced or not. The one last thing I want to say about their production is that typically these peptide hormones, especially the peptides, are synthesized, let's say right here, this newly synthesized one, as a prohormone. What does this mean? This means hormone. It's actually synthesized, made up of several different pieces. So I'm trying to draw these different colors here to represent the prohormone. What's going to happen is in the vesicle, as part of the secretion and ultimately final steps of production of this hormone, it's going to be chopped up. These are scissors. Can you tell? Scissors. This process is regulated. So the prohormone is going to be chopped into its final products. So then in the end, we've got, in this case that I made up, we have 3 different, that's the wrong one over here. 3 different final hormones produced from one gene. So they're regulated kind of together. So just to know that these are produced first as prohormones that need to be further processed. We won't look at this a whole lot more, but ACTH is actually an example of this. Okay, last thing for peptide hormones is their action on the cell. So clearly these big old things cannot go through the cell membrane. They're going to bind to membrane receptors, and this is where I want to detail a little bit more, reminding you the steps in a G protein coupled receptor. And typically, this is the pathway we'll see. So CAMP is the second messenger. Sorry, that's a really light color. So we've got a G protein that is activated by the first messenger, the hormone binding. The G protein is going to activate adenylyl cyclase. Don't really need to worry about this thing. Adenylyl cyclase is going to convert ATP to CAMP. Here's our second messenger. The other second messenger we have seen, and we'll see briefly again, I think, is calcium can act as a second messenger, which just means the second messenger in this cascade. This is the messenger that can act inside the cell. It can do things like activate protein kinase. This is one example of what it can do. CAMP could do potentially other things. It's converting the message from outside of the cell to inside the cell, cellular transduction. Protein kinase can activate, phosphorylate other proteins to activate them. So this has all kinds of effects on the cell. Last but not least, we've got amino acid derivatives. I already told you what these were in the basic idea. Here's another example of their different types here. Oops, I just drew on there. So, and we will look at these three types in different detail. So they're all derived from amino acids. Thyroid hormones, one big category. Catecholamines, these are all shown down here. They are derived, did I say tyrosine before? Oh yeah, there's tyrosine, yeah. Phenylalanine and tyrosine shown down there, so derived from amino acids. And then the other one is a tryptophan derivative. Tryptophan is the food, the amino acid found in turkey that is blamed for your post-food coma at Thanksgiving. It's the precursor to melatonin and also serotonin. We won't talk a whole lot more about serotonin, these two, but I do want you to be able to have them in your mind as in this category of amino acid derivatives. Otherwise, we will actually focus, we'll talk a little bit more about thyroid hormone when we talk about the thyroid. Catecholamines will come up quite a bit. You know epinephrine already. You know norepinephrine. Epinephrine and norepinephrine are neurotransmitters. Norepinephrine is a very common neurotransmitter in the autonomic nervous system. Both epinephrine and norepinephrine can act as hormones when they're released from the adrenal medulla. So we'll see them again there. You may have also heard of dopamine, which is a neurotransmitter. Wait a minute, why am I neurotransmitter hormone, what? Remember, it can be the same molecules. So norepinephrine is a hormone when it's in the bloodstream, and it's a neurotransmitter when it's released into a synapse. Dopamine is primarily a neurotransmitter. Epinephrine is primarily a hormone. Okay, these all bind to cellular membrane receptors, receptors on the membrane, except for thyroid hormone. So these two are going to bind to membrane receptors. What does that mean for their function? They're probably going to activate G protein coupled receptors. And that's what you've actually seen some. You saw in the fall, norepinephrine binding to, both epinephrine and norepinephrine binding to alpha and beta receptors. Remember those? Those were G protein coupled receptors. Thyroid hormones are going to be carried into the cell to bind to intracellular receptors. And that's because they have a carrier protein that helps them in. So we'll look at, so they're actually going to alter gene expression as well. That'll be important for thyroid function. Anything else here? Okay, here is a quick review with some nice images. this is a nice image showing the two different types of receptors. Cell surface receptor, right? This is the same thing as a membrane receptor. And here it tells you what binds to those, what types of hormones. A nuclear receptor, wait a minute, what's that? That's an intracellular receptor. Why is it also called a nuclear receptor? Because it's going to go into the nucleus, ultimately. And that's steroid and thyroid hormones.